our file. So these are the identifiers for those samples on this column. Here are our genes. They're on these. This is the Affymetrix identification. So if you went to Affymetrix and you looked up this identifier for this chip, you could figure out what gene that is. We are going to tell the software what this is. Don't worry about that. Okay? But you can see, and you get down here, so we've got 30 samples, and we're probing about approximately 36,000 genes. Is everybody with me so far? This is simple, isn't it? It is. Everybody's looking at me like, it's going to get harder, I know it. No, it's not. It's not going to get hard. It's pretty easy. Okay? So we're at this point. What are we missing? What do we need to do to this now? What do you think? Categories, right? We don't know what the samples are. Right? Where do you think you're going to get that information? Yeah, Geo, totally. Actually, we're not going to get it from Geo because Geo sucks. We're going to go to the European version. They're much nicer. They drink tea and you're just happier people. Geo's horrible. Okay. So we're going to need that. We're going to need a sample table, right? We're going to need to take that information and merge it to this, correct? I'm going to teach you how to do that today. What other information do we know? Do you want to see this information? Would you rather see genes up here? Totally. Okay, and number 2 is we're going to get rid of these 2 columns that don't have anything in it. So I'm going to hit, and this, honestly, Partek works just like Excel almost. So I select these 2 columns, I right click, I go delete. Bam. I will say, and I'm going to tell you right now, you've officially got your warning, there is no backspace in Partek. None. Just to warn you. Man, I've been stuck on Excel, so, like, one time I hit, like, backspace like 14 times just to get to where I needed, like, because I messed up. So I had to hit, but I recovered it. But when you mess up here, it stays. So no backspace. All right. Well, let's tell the software what this is. Okay? So I am going to go, so you'll see our file names up here, the GSE 27087, right? Okay. Make sure it's highlighted, right click it. Hit properties. Okay, everybody see this window? Okay, I'm going to say that this is a genomic microarray. Trust me, it is. Okay. This is another cool part. So you can imagine, you've got 33,000 things. You've got to find a file that basically tells this software what that is, an annotation file. Normally, before, you'd have to go to Geo, and then you'd have to download their annotation file, and then you'd put it somewhere, and then you would upload it here, and it would take you about an hour. I kid you not. Here's what we're going to do. Is if you go down here, we've got the genomic data, marker ID and com. We don't have to worry about this. This is where it's telling us what chip is this. Basically, it wants to know what it's trying to look at. You can download that annotation file, and usually you get that from Affymetrix. Here, what we can do is, since Affymetrix is a pretty popular company, Partek keeps their annotation files on file at their website. So go to download. Everybody hit download? Okay. Tell me what that chip was. Somebody go to the original Geo entry and tell me what the chip is. Yep. Okay. So we go here, click on this. We want to find mouse. Actually, I think it might be Mo. I'm not sure. Wait, ooh. Oh, there, mouse gene. Okay, mouse gene. 1.0, and here's what I want you to do. Go gene analysis. This is the one we want. You could do the other ones, but I think gene analysis works better, and I'll tell you why later if you ever want to know. Hit OK. All right, species. And do you see how it filled in everything here? So now we have this on file. If I did another chip using this, did another analysis using the same kind of chip, I wouldn't have to download this anymore. OK, hit OK. And hit OK. All right. Here's what I want you to do. Once you've downloaded it, it hasn't saved it. We want to save this information, so hit Save. See this? You can do it under File, or there's got this, looks like a little disk here up here. Hit Save. Now that annotation information is attached to all these numbers. This software now knows that 10338001 is, say, gene A. And it knows that for all 35,000 of these things. Okay? Now, as you mentioned, there's no categories, right? We got all this pretty expression data, but we don't have any way to analyze it. We have no comparisons. Yeah.
Is your array file, annotation file, or downloaded pretty much?
Is it really?
Yeah.
Okay. Give it a minute. In the meantime, so here's the deal. So in the meantime, we can do this.